So we're now in chapter 24, which is nutrition, metabolism, and energy balance. And let's begin. So let's start with nutrition. Nutrition is actually the study of the things that we actually eat, and these are the nutrients that is needed so that we can stay healthy. Most nutrients are used for metabolic fuel, but some are for cell structures and molecular synthesis. So what is exactly a nutrient? A nutrient is a substance in food that's needed for growth, maintenance, repair, and there are two types, or they fall into two categories. One is what you call the macronutrients, and the other one is your micronutrients. Macronutrients are made up the three major nutrients that make up the bulk of our ingested food. These will be your carbohydrates, your proteins, and your fats. The micronutrients and pretty much are two other nutrients that are required, but only in small amounts. These will be your vitamins and minerals. And then there's also water. Water is required for us to stay alive and healthy. And so water is technically a nutrient. So non-essential nutrients are also vital in life, but it's not enough. But if not enough of this is available, the liver can usually convert other nutrients into what is needed. That's why you call it non-essential. It does not necessarily we do not necessarily need to put it in our diet because our own body can produce it. Now, the energy value that we're going to use as we talk about nutrition and also metabolism, etc., is actually calories, kilocalories. 1 calorie is the amount of heat that's needed to raise a temperature of what 1 kilogram of water by 1 degree Celsius. And one dietary calorie is equal to a kilocalorie or 1,000 calories. And right here, we do not have the USDA's My Plate. It's actually a pyramid. I still would prefer to go to the pyramid, I'd say, because a pyramid pretty much is a good representation of amount. And so I could see that right there, the tip of the pyramid, you have, um, well, this exactly the entire things or food that is actually inside the pyramid is what we are supposed to be eating per day, and we use sparingly the fats and oils. It's at the tip of the pyramid. Then the one that's actually right here at the bottom, which is the widest portion of the pyramid, will be your bread, cereal, rice, pasta group. This is uh, kind of like cheaper, and pretty much it is actually used um much more, most especially by growing kids, because this is actually the one that gives them energy. Then as we go forward, vegetable with vegetables, we require only about 3 to 5 servings per day. Your fruit groups is actually 2 to 4 servings per day. Your milk and yogurt is actually 2 to 3 servings per day. And your milk and poultry, fish, dry beans, or proteins, heavy proteins, fats and but not fatty, because the fat is all the way up. It's 2 to 3 servings. Remember that these will have also fat, and you can see that the fat will be those yellow buttons right there, and the white buttons will be sugar. So the fat is almost like in every food that we're taking. So why do we need to eat fat? We don't really need to ingest it because it's there. And this is actually, it would look like there's too much per day for one person to eat. Well, this is actually the plate of a growing active individual. As you as you grow and change your way of life and you get less active, then you will have to make sure we will have to make sure that we will have to make adjustment in the amount of food that we are taking, and we should also be don't forget that we should also be exercising in order to live a well balanced life. So let's take a look at this carbohydrates, lipids, and proteins before we get into metabolism. Carbohydrates pretty much is primarily from plants, such as starch, which is actually your complex carbohydrates, and there's also in grains and a lot in vegetable. Now, carbohydrate is made up of sugars itself. You have your monosaccharides and your disaccharides. And the sugars in fruits, sugar cane, sugar beets, honey, and milk. Okay, these are the sugars. Then you have also the carbohydrates with insoluble fibers. The cellulose in vegetables pretty much are the ones that provide roughage. Then you have the soluble fibers of pectin in apples and citrus fruits. They reduce blood cholesterol level. We find a small amount of this in soluble fiber in milk and in milk sugar, and glycogen in meats. Now, what's the use of these carbohydrates? Well, the use of these carbohydrates is actually that just one molecule that is the unit for carbohydrates. It's actually glucose. Fuel that is used by the body, which is ATP, is made by the body from glucose. But there are also some, there's also a good amount of ATP that we use from fats. And neurons and red blood cells rely entirely on glucose. Neurons die quickly without glucose. But excess glucose is converted to glycogen or fat and then stored. And that's how we gain weight. Fructose and galactose are converted to glucose by the liver before they enter circulation. So our carbohydrates is stored in the body as glycogen in the muscles and in the liver. And depending on how active we are, if we exercise daily, or the type and intensity of our of our general movement, that's pretty much will spell out how much we need, of how much energy we will need, and that is actually calling for a carbohydrate diet. Now, so we get our carbohydrates from the sugars and the starches. Pretty much, it helps us in recovery from required performance and from training, and a depletion of carbohydrates in the body would result in glycogen depletion, and this could result in fatigue, getting tired, most especially when we exercise. So, what will be the function of the carbs? Well, just like what I mentioned, the unit for the carbohydrate that is necessary in the production of ATP is glucose. And every complex carbohydrate, and of course a simple carbohydrate, would eventually be converted to glucose, which is the unit. And when we digest it, chemically break it down, we absorb it as glucose or other monosaccharides that is absorbable through the gut. Now, the daily recommended intake of carbohydrate is about 45 to 65% of your total calories. And the diet should consist mostly of complex carbohydrate, not sugars. Simple carbohydrates should be limited. High amounts of sugars can lead to obesity, because then you have already like a good amount of glucose that is already available to be converted into ATP. And if there's going to be too much of this high energy molecules around and you are not really using the energy, then you are going to be converting these carbs into fats. But the American diet is at roughly around 46% carbohydrates, and that includes our our obsession with rice and pasta or breads. As a matter of fact, from the country where I came from, a day without my uncle used to say, a day without rice is a day without sunshine. Oh darn, it is really. Rice is our pasta, and that's in the Philippines. So uh, these carbohydrates tend to be inexpensive, and therefore there's higher percentages in developing countries and low income groups. This is a very good summary, a table that will guide you in your studies of carbohydrates, lipids, and proteins. There's going to be another table later on that I can show. This is only for carbohydrates. So now we get into fat. And see here, I'm showing you the foods that are high in fat. And the dietary sources pretty much of, well, fats is actually triglyceride, which is animal fat, and they're found in saturated fats, in meat, dairy foods, tropical oils or hydrogenated oils, and the unsaturated fats are found in seeds, nuts, oils, and most vegetable oils such as avocado oil. And cholesterol is found really heavily in egg yolk, in red meats, organ meats, shellfish, and milk products. And pretty much, uh, we don't need to take cholesterol. We don't need to load ourselves with these sources of cholesterol because our own liver makes 85% of the cholesterol that we have in our body. So stay away from cholesterol. Then what are the uses of adipose tissues? Well, adipose tissue offers protection and insulation and fuel storage. Protection because they are the ones that are covering and insulating and protecting our muscles and our bones and our internal organs from harm. You have the fatty covering of our body is the one that is protecting it from mechanical injury. But there are other types of fat that would have different what do you call this, purposes. Like phospholipids is essential in myelin sheath and cell membranes. Cholesterol stabilizes membranes and is a precursor of bile salts and steroid hormones. Matter of fact, you should know that your own has estrogen and your testosterone and some and other what do you call this, steroid hormones are actually cholesterol based. So the they start from cholesterol and our body produces it and turns it into estrogen and testosterone and whatever. Prostaglandins are the chemicals for inflammation. They're also necessary in smooth muscle contraction and blood pressure control. And of course, fat gets converted to ATP. It's a major fuel of hepatocytes and skeletal muscles. And they help also absorb fat soluble vitamins in the intestines. Fat should represent only about 20% to 35% of total calorie intake. Saturated fat should be limited to about 10% only. And cholesterol, we should not then would take it. If at all, we could actually get away from it because our own liver produces cholesterol. This is the other summary, which is actually this one is telling us something about the fats, which is actually just to summarize what I just mentioned. Then we get into proteins. Proteins come from animal products, eggs, milk, fish, mostly meats, as well as soybeans are considered to be complete proteins because they carry all the essential amino acids. So how many amino acids is actually needed when you say all amino acids? Well, the human protein is actually made up of 20 amino acids. So the way our human protein is actually like formed in the cells is that it follows what you call an all or none or all nothing principle, which means you have to have all of the raw materials, all of the amino acids right there, so you can produce that protein, or that protein will not be produced at all. So if you're missing one or two amino acids to produce a single protein in a human body, that protein will not be produced if your two missing amino acids will not be replaced. There cannot be any way of producing this not unless you have all of the raw materials right there. So what's going to happen to the amino acids that is supposed to be lined up to become this protein? Well, they're all going to be burned down to become like a source of energy, or converted to fats, or converted into other macronutrients such as carbohydrate. So um, we all contain all the needed essential amino acids, pretty much, or um, the things that supposedly we cannot produce and therefore we need it in our diet. We call that essential amino acids. And we have 9 essential amino acids. And the 9 essential amino acids pretty much is needed to be part of our diet because we cannot produce them. And there's 9 of them. So here is actually some sort of a Venn diagram showing you the amino acids, the non-essential and the essential. We have your 9 essential amino acids. 1, 2, 3, 4, 5, 6, 7, 8, 9. So you have all the essential amino acids. You cannot you cannot actually miss this 9. If you miss one of this, our body will not be able to produce one of this, and the protein that where that amino acid is required will not be produced, and you're going to be malnourished. Now, the non-essential amino acids is actually the amino acids that we pretty much produce in our body, and therefore they are not essential in the diet. The ones that is in the middle is what you call conditionally essential, which means in certain conditions, or it's at certain times where we are actually very active and we are burning and we need more of the amino acids to form our muscles, then we need these non-essentials become essential, which means for our body to replace the tissues, you will need to have this also in your diet. But this, the alanine aspartic acid, asparagine, as well as glutamic acid, this is actually pretty much really always non-essential. All right, even in times where we are under stress, or we are actually like producing a lot of muscles, these two we can produce a lot of them. These four rather. So let's actually ask a question. Can a vegan diet provide all essential amino acids? Well, the answer is yeah. A well-planned vegan diet can supply all 9 essential amino acids, though pretty much it requires consuming a varied selection of plant-based foods to ensure adequate intake of all essential amino acids. As different plants, different sources have varying amounts of them. You don't need to worry about combining specific foods in the same meal as when you are eating like the meat. You eat the chicken and everything, all amino acids is already there. Or you're eating an egg, all the amino acids are already there. So the body pulls amino acid from a variety of sources, or a variety of food that you've eaten throughout the day, and that's pretty much where we get the amino acids to produce the proteins that we are in need of for that particular day. All essential amino acids are present in plant-based foods, but the concentrations will vary. Some plants would have only 18 amino acids, like your corn will have 18 amino acids, and then your beans probably would be pretty much like there are beans that would carry also 18, but a different set of 18, not the same 18 as a corn. And there are some beans that actually that are considered already like complete food. They carry all 20 amino acids. So so what are the uses of proteins in the body? Well, first of all, um, um, structural materials. Proteins are needed pretty much. This I consider this also structural, which means, example, for instance, your keratin, your collagen, your elastin. These are all connective tissues, and the muscle proteins. They're all made up of proteins. Collagen is protein. And then the functional molecules, the molecules that is um, uh, well, the functional molecules that move your muscles, your antibodies is a functional molecule. So pretty much these are, and enzymes are also functional molecules. These are, and some hormones, these are proteins that, these are substances that are actually proteins. And there are three factors that would help determine whether amino acids are used to synthesize proteins or really be burned as a fuel. The first factor is actually the all or none principle. All amino acids needed must be present for protein synthesis, just like what I mentioned, or not at all present. Then the amino acids are actually diverted into energy consumption. The adequacy of calorie intake. Protein is used as fuel if insufficient carbohydrate or fat is available. Then hormonal control is another factor. The anabolic hormones such as growth hormone and sex hormone accelerate protein synthesis and growth. The adrenal glucocorticoids that is released during stress promote protein breakdown and conversion of amino acids to glucose. So pretty much these are the factors that help determine whether amino acids are used to synthesize protein or burned as fuel. So if the amino acid is not converted into protein, they will be burned as fuel. So we have here an amino acid pool. Let's take a look at that further. You have here an amino acid pool. And in this amino acid pool, your body protein gets to be degraded and becomes amino acid. The dietary proteins that we eat after digestion and absorption, most of them it gets into the amino acid pool. Then you have the synthesis of non-essential amino acid. All of them gets into the amino acid pool. Your amino acid pool is used by the body in different ways. They are used by the body to synthesize our own protein. They are used by our body to synthesize the non-protein nutrients, such as your creatinine, hormones, neurotransmitters, purines, pyrimidines, niacin, thyroxine. These are nutrients, and these are also part of our body. But we need amino acid to um, make all of this. Then some of them gets to be broken down, okay, and in catabolism itself, these are actually used or converted into glucose and glycogen in the liver. They're actually going to be its breakdown would result in the formation of urea, carbon dioxide, and water, and then of course ketone bodies and fatty acid. This is actually what the amino acid, our amino acid will actually turn into. From the time that we ingest them or we get them from somewhere else, they pretty much like get into a pool. If they're not all going to be coming into becoming a part of our body protein, they're going to be used somewhere else by the body. And here is actually the third table, the third part of the table, which is you can use for your study. And this now will discuss the proteins. So now let's get to the role of vitamins and minerals. So, excuse me. Vitamins are organic compounds that are very crucial in helping the body use the nutrients that we take in. And most of the vitamins function as coenzymes. Most of them must be ingested, except for vitamin D, which is actually made in the skin. We already have our vitamin D. Some vitamin B and K are synthesized by the intestinal bacteria. The beta carotene, the ones that are from carrots, is actually converted in the body to vitamin A. There's no food group that contains all vitamins in in that one particular food product. So there are two types of vitamins based on their solubility in water. One is what you call the water soluble vitamins, and the other one is the fat soluble vitamins. The water soluble vitamins are the B complex and the C complex. The B complex and the C complex are absorbed with water. B12 is actually absorbed through the gut, but it requires the intrinsic factor. They're not stored in the body. These water soluble vitamins are not stored in the body. If any that are not used within the hour, they are excreted pretty much. So you take your vitamin B and you take your vitamin C within the hour. You go to the bathroom and you see them all in the bathroom. Your urine is going to change in color. So you excrete them within the hour. Now, fat soluble vitamin though is different. These are your A, D, E, K, and they're absorbed with lipid digestion products. They're stored in the body except for vitamin K, and excessive consumption can cause actually health problems if you are consuming too much. Okay. And here's another table, which is actually for vitamins. It can help you um, review the vitamins. Now we go to the minerals. There are only 7 minerals, and there's 8 right here in the dietary minerals. These are part of our dietary minerals, but only 7 are required in moderate amounts: calcium, phosphorus, potassium, sulfur, sodium, chlorine, and magnesium, and are required in trace amounts. The others are required in trace amounts, and pretty much all of this is actually to ensure proper functioning of metabolic activities. The uptake and excretion of dietary minerals are balanced to prevent toxic overload. For instance, they are really very useful. For instance, calcium, phosphorus, okay, and magnesium. mm, These are salts that harden the bone. There are parts of the bone that makes it hard. Iron is essential for oxygen binding to hemoglobin, and iodine is necessary for thyroid hormone synthesis. And sodium and chloride are major electrolytes in our blood. The, so we need the minerals, and mineral rich foods will be your legumes, your vegetables, milk, and some meats. And here's another table of dietary minerals, so you will know exactly what they are and major sources, major function in the body, and if you have a deficiency, what will be your symptoms. So now we go to the biochemistry, and we will start with discussing metabolism. This is really going to be really a lot of chemistry, guys. So I'm going to discuss it with as much as possible by not using too much of chemistry, because this is not a biochemistry class, it's an anatomy class. But you will need to really know exactly what or how the body works around with its food, and you will need to know a little bit of the chemistry of metabolism. So, what is metabolism? Metabolism is the sum of all the chemical reactions in the body, and there are two types of metabolism. One is catabolism, and the other one is anabolism. Catabolism, you see here a big molecule, and it's broken down into its smaller units, and there is actually the use of energy. In anabolism, you require energy and pretty much the smaller molecule to build molecules. So anabolism is a set of metabolic pathway that would construct the molecule from smaller units into a bigger unit. Meanwhile, your catabolism is a set of metabolic pathway that breaks down molecules into smaller units that are either oxidized to release energy or used in other anabolic reactions. So, cellular respiration. Now we go into more of biochemistry. Cellular respiration is the catabolic breakdown of food fuels, where energy from food is captured to form ATP in cells. And the goal of cellular respiration is to trap chemical energy in ATP. Energy can also be stored in glycogen and fats, which can be broken down later. Phosphorylation is another metabolic reaction. Here, the enzymes shift high energy phosphate groups of ATP to other molecules. Phosphorylated molecules become activated to perform cellular functions. So, here, cellular respiration, the aim of cellular respiration is to capture the energy in glucose and convert it into ATP. And the purpose of your phosphorylation, this second metabolic reaction, is to capture the energy in ATP and pretty much phosphorylate the molecules. And when molecules are phosphorylated, they're activated to perform cellular functions. So it's actually one molecule into another molecule that's high in energy, and this high energy molecule phosphorylating because ATP has a phosphate group, so it could freely release that phosphate group to energize another molecule and form another compound. So what are the stages in processing a nutrient? So we ate food. What happens to our food? There are three. You have stage one, pretty much, and that is in the gastrointestinal tract right here. Nutrients are digested into their absorbable units. The absorbable units of your protein is your amino acid. Your carbohydrates, the absorbable product will be glucose and maybe galactose and fructose. That are all monosaccharides. And then the fats and oil need to be broken down into glycerol and fatty acids before they're broken down. Once they are broken down and then absorbed, they are absorbed in the tissue cells. Here, they're going to be broken down. There's going to be either anabolism or catabolism. So the amino acids are now going to be put together to form protein. That's actually anabolic. And here, the glucose and other sugars in the liver will be put together to form a bigger sugar, which is actually found in our muscles and in our liver, which is called glycogen. Then again, fats and glycerol are put together in our liver to form triglycerides, which is our own animal fat. But then, amino acids in catabolism, nutrients are broken down, okay, into pyruvic acid, the acetyl coenzyme. Can take a look at that. The sugar here is broken down into pyruvic acid and into acetyl coenzyme, which gets into what it calls the recycling of energy to form new energy, high energy molecule in the Krebs cycle or in the citric acid cycle. And pretty much these are all coming from glucose and fats itself, the acetyl group of the fat. So that is actually all, that's actually in the tissue level. In the this particular citric acid cycle, this is not happening just in the tissue, this is happening in the cells itself, in the mitochondria, inside the mitochondria of the cells. There are oxidative breakdown, pretty much of stage 2 products, the products right here, okay? This is where carbon dioxide is released, okay? And water is also produced here. Carbon dioxide is that the carbon dioxide that is produced here in cellular respiration is exactly the carbon dioxide that we breathe out. Imagine doing cellular respiration in every single cell, in every single mitochondria. And how many mitochondria do you find in a single cell? Many. So you have many mitochondrial cellular respiration happening in all of the cells in the body, okay, except for the red blood cells, because the red blood cells don't have any mitochondria. So every single cell in the body is actually producing carbon dioxide, that the gas that we exhale. And at the same time, they're also producing ATP. So we're going to get into those, the ATP production in a short while. But first, let's take a look at some, what do you call it, some, a few of the reactions and the names. Semantics. Let's take a look at some semantics. Oxidation reaction. This involves the gain of oxygen or the loss of hydrogen atoms and their electrons. And you call these the oxidation reduction reactions, or in chemistry we call it redox. Oxidized substances would lose electrons and energy. Reduced substances would gain electrons and energy. So redox reactions are catalyzed by enzymes that usually require vitamin B coenzyme. And these are what you call the dehydrogenases. They catalyze the removal of hydrogen atoms if hydrogen atoms need to be removed, and that's actually during oxidation. And oxidases catalyze the transfer of oxygen. And there are two important coenzymes that act as hydrogen or electron acceptors in oxidative pathway. And this will be your nicotinamide adenine dinucleotide. These are very important molecules, guys. This will be in your test, so remember that, please. And flavin adenine dinucleotide. These coenzymes will be energized um, in what do you call this, in the reactions that we're going to see to produce NADH and FADH2, which is actually the high energy molecules. These are the low energy form of the NADs and FADs. When they become, when they get into the Krebs cycle, the NAD becomes NADH, which is a high energy molecule. The FAD becomes FADH2, which is another high energy molecule. And we're going to discuss that in just a few seconds. Okay, here's another word that you need to know, that you need to understand the next few pages. Two mechanisms are used to make ATP from captured energy, and that is liberated during cellular respiration. The first one is what you call substrate level phosphorylation. This is substrate level phosphorylation. This is where your high energy phosphate groups are directly transferred from they are actually transferred from the phosphorylated substrates to ADP and forming ATP. This occurs in glycolysis and in the Krebs cycle. The necessary enzymes here are in the cytosol. That's why glycolysis happens in the cytosol, the cytoplasm, and also in the mitochondria for the Krebs cycle. This is the second type of phosphorylation. This is actually what you call, the first one was substrate level phosphorylation. The second one is oxidative phosphorylation. Oxidative phosphorylation is a more complex process, but produces most ATP. And in a process which is called chemiosmosis or chemiosmosis, this particular process couples movement of substances across the membrane to chemical reactions, and energy is released from oxidation of food and is used to pump hydrogen ion across the inner mitochondrial membrane, creating an gradient. I will show you exactly where the gradient is when we get into the actual site in cellular respiration where oxidative phosphorylation is happening. Just so you will understand that in oxidative phosphorylation, there is a creation of protein, or rather, uh, um, proton gradient. And the proton gradient is when a lot of because of phosphorylation, oxidative phosphorylation, high energy molecules will pump in hydrogen ions on one side in the intermembranous space of the mitochondria, making it concentrated, highly concentrated in hydrogen ions and much more concentrated than in the mitochondrial matrix. This is actually the mitochondrial matrix below that. Remember that this is your inner membrane of the mitochondria. And so there is a gradient between them. And when there is a gradient, those molecules there that are in the other side would want to get out of there and travel from an area where there's too much of them towards an area where there's less of them. And the only way to go is when there's actually an opening, which is actually your ATP synthase. So all of these hydrogen ions move through here. As they are moving through here, they are actually making the ATP synthase convert ADP to ATP, which is really cool. I'm going to show you exactly where you find this oxidative phosphorylation in cellular respiration in a moment. Give me a moment first. Okay, so this is our carbohydrates. We're now going to get into metabolism of carbohydrates. So in the metabolism of carbohydrates, remember that we start with glucose because this is the absorbed state. And um, in the body, glucose plus the oxygen that we breathe will now be converted into water plus carbon dioxide and 32 ATP plus heat. This is actually the formula for cellular respiration in our body. So the glucose that we ate from the sugar or from our dietary carbohydrate sources, plus the oxygen that we breathe together during cellular respiration will form water plus carbon dioxide plus 32 ATP. So it is phosphorylated. When glucose enters the cell, it is immediately phosphorylated into glucose 6 phosphate. This is actually when glycolysis happens in the cytoplasm. This glucose will be broken down, and the first intermediate compound that it will become will be a glucose 6 phosphate. And glucose 6 phosphates pretty much would move forward again on the process of glycolysis into fructose 6 phosphates and then eventually into pyruvate, which is the byproduct of glycolysis. This is the one that gets into the Krebs cycle. And um, some of our absorbed monosaccharide, which is not glucose, and those galactose and fructose, these are not glucose, needs to be converted into glucose 6 phosphates and fructose 6 phosphates so that they can also participate in the production of ATP. They are all converted into pyruvates. But if they are not going to be burned and they will not help in the production of ATP, in the liver they are going to be used in the production and they will be stored as animal fat. And in animal fat, we call that animal fat glycogen. That's actually stored in the liver. Okay, so the complete glucose catabolism would require three pathways. And these three pathways are actually right there in front of you. You have your glycolysis. This is the first pathway. Then you have the Krebs cycle, second pathway. Then you have your electron transport chain, pretty much. Glycolysis is the breakdown of glucose and the cytoplasm. Remember that it is in the cytoplasm. The rest of them happens in the mitochondria. This is the mitochondria. It is one of the organelles inside the cell. Now, glycolysis happens in the cytoplasm and would break down each glucose molecule into two molecules of pyruvic acid. The pyruvic acid enters the mitochondria and will pretty much like enter the Krebs cycle. The pyruvic acid then will be entering the what do you call this, a cycle of um, a cycle of chemical reaction, where citric acid is actually oxidized to form more carbon dioxide and high energy molecules and ATP. A small amount of ATP is produced here, but large amounts of NADH and FADH2, which happens to be the energized form of NAD and FAD. So this, or there's actually the third pathway, which is the electron transport chain. Byproducts of the citric acid, the high energy molecule FADH and NADH, FA, FADH2 and NADH2, NADH will now be getting into the electron transport chain and will produce roughly about uh, 30 to 34, um, or if you want the stereotype calculations, theoretically, they can actually produce up to 36 to 38 ATP, depending on who's calculating it and what they're using for calculation factor. Okay, so let's take a look at glycolysis. Glycolysis is also known as the glycolytic pathway. It involves a 10-step pathway. Um, it is anaerobic. It occurs even with the absence of oxygen. It happens in the cytoplasm, and glucose is broken down into two pyruvic molecules with three phases, as I mentioned, that the sugar activation, the sugar cleavage, and the sugar oxidation and ATP production. Here is your glycolysis. In the phase one, which is the sugar activation, this is where your glucose is going to be activated by addition of two phosphate groups on each end. So now it becomes a fructose 1,6 bisphosphate. Where do you get the two phosphate group? Well, you break down two pieces of ATP, and this will phosphorylate the two ends of the glucose. So you lost two ATP, but you're not supposed to lose ATP. I thought your glucose is going to give you energy. Well, this is only an investment. Remember, when you go to the casino, you want to win something, you have to invest your money, right? And that's when you end up really winning something. And the same thing is happening here. You're investing 2 ATP with the hope that you're going to get more ATP. As a matter of fact, this is really more ATP because you only invested 2 ATP and you'll end up having 38 ATP in the end, which is really rich. Oh my goodness. So, second phase is when your 1,6 bisphosphate is now going to be cleaved. Okay, the cleaving of the sugar is the second phase. It is cleaved into two 3-carbon fragments. One is actually the G3P, and the other one, well, actually, these are isomers. This one is called dihydroacetone phosphate. This one is the glyceraldehyde 3-phosphate, but they are isomeric. And finally, in the third phase, which is the oxidation of sugar, this is where you produce two molecules of pyruvic acid and you form 4 molecules of ATP. Wonderful. Now you're seeing ATP. But you used up 2 ATP in the first phase, so the net production, ATP production for glycolysis is only 2, really. And of course, you take up one low energy NAD and you convert it into two high energy NADH, which is really good. Now you're seeing high energy molecule and you're seeing ATP. This is the end of your glycolysis, because now your pyruvic acid will now have to get ready to get into the citric acid cycle. And this is actually the 3 major phases. If you want to go over and take a look again, I will leave that to you. Now, let's take a look at the citric acid cycle. This is also known as the Krebs cycle. It was named after Hans Krebs. Excuse me. Hans Krebs is the scientist that pretty much developed the citric acid cycle. He worked all his life in trying to figure out how this particular cycle uh, is happening. He discovered that this particular cycle is very acidic. As a matter of fact, he called it the citric acid cycle because it seems that it is always producing citric acid. That's why he called it citric acid cycle. Upon his death, the citric acid cycle was named after him, and we call it now the Krebs cycle. This Krebs cycle occurs in the mitochondrial matrix. It is fueled by the pyruvic acid from glucose, which is actually the byproduct of glucose breakdown, and the fatty acid also from fat breakdown. Pyruvic acid cannot go as is inside the citric cycle, the citric acid cycle. It cannot enter. It has to be, the citric acid cycle does not accept the pyruvic because it is actually a 3-carbon compound. They need only the acetyl or 2-carbon compound coming from there. So they need to remove, and that pyruvic acid will undergo a transition reaction, a transitional phase, where the carbon is going to be removed. And in that transitional phase, you will need an enzyme also to pick up the acetyl, that particular 2-carbon, and then that 2-carbon will be picked up and delivered by the coenzyme into the citric acid cycle. So what happens to that carbon that's been removed from the 3-carbon pyruvic? It becomes our first molecule of carbon dioxide. And the same token, in this transition phase, for every 1 pyruvate, you also produce 1 NAD, and therefore in the 2 pyruvate molecule that is coming from glycolysis, you will have 2 carbon dioxide as byproduct and 2 NADH as byproduct. Now let's take a look at your what happens to the pyruvic acid, rather to the, not the pyruvic acid, but to the acetyl coenzyme. We're done with the pyruvic acid, so the remaining 2-carbon fragment, okay, that is oxidized into acetic acid by the removal of hydrogen, will now be picked up by NAD. Okay, it will now be picked up by NAD, and it will form there's your NAD right there. It will form your acetyl coenzyme, and the acetyl coenzyme pretty much is actually the one that is going to merge into the citric acid cycle. And this is actually just in the transition reaction. Pretty much the coenzyme is only a shuttle. The coenzyme A shuttles its acetic acid to an enzyme in the citric acid cycle to join it, and it's actually going to be joining a 4-carbon intermediate, which is of oxaloacetic acid, and it will become a 6-carbon intermediate, which you call the citric acid. It produces a citric acid, and this is why it's called the citric acid cycle. Now, during the 8 steps of the citric acid cycle, the original acetic acid is pretty much like decarboxylated and oxidized into a keto acid. And the intermediate reaction produces NADH. NADH. Nope, I'm not pointing to that. I'm pointing to this NADH and FADH2. Let me see. There, that's also your FDH. So NADH plus FADH, and you have other NADH here also. In the final step of this entire sequence of cycle will be again the formation of oxaloacetic acid. And once you have oxaloacetic acid there, it's going to wait for another couple of acetyl coenzyme A to shuttle in the acetyl group. And then it becomes citric acid again, and then it will go again and form NADH. There's actually 3 NADH that's going to be forming here, 2 FADH. I mean, 1 FADH for every turn. And then for every turn, you have 2 carbon dioxide that's going to be formed. But 1 turn is only for 1 pyruvic acid. And there are always 2 pyruvic acid coming from 1 glucose for every glucose molecule, therefore. There's going to be in Krebs cycle the formation of 2 NAD, the formation of 1, 2, 3 times 2 is 6 NADH, and then 1 times 2, 2 FADH, 2 ATP, 2 ATP is also going to be formed, and 4 carbon dioxide in the citric acid cycle. So all in all, in between your transition reaction and your Krebs cycle, your carbon dioxide, the total carbon dioxide molecule that's formed is actually 6, not 3. You have to multiply that by 2 because there are 2 pyruvic acid that pretty much is coming from the breakdown of glucose. So now, what happens to this high energy molecule? You have high energy molecule NADH, you have 6 of them. You have 2 FADH2. You have also ATP from glycolysis, and you have ATP also from your Krebs cycle. They're very little, there's only 4 of them, but there's a lot of NADH and FADH2. Now, NADH and FADH2 pretty much is the main player, one of the main players in delivering energy into the electron transport chain and oxidative phosphorylation. Through oxidative phosphorylation, these will actually be they will be actually producing more ATP. For every one NADH2, there's actually a factor that would convert one NADH2 to about 2.5 to 3 ATP. And then the FADH2, there's actually the factors that would be converting FADH2 to about 1.5 to 2 ATP. So you could just imagine for every one NADH, 3 ATP can be produced. For every one FADH2, there's actually a chance of producing 2 ATP. That's a lot. And because you have produced a lot of FADH2 and NADH2, you are going to be looking forward into a lot more ATP than just 4 ATP. So, how does it happen? Well, the electron transport chain creates proton gradient across the mitochondrial membrane using high energy electrons, which is actually your NADH and FADH2, and are all coming from on the Krebs cycle. Then it undergoes chemiosmosis. Chemiosmosis uses the energy of the proton gradient to synthesize ATP. The proton gradient proton happens when hydrogen ion moves into the intermembrane space from leaving a great difference in concentration of proton between the intermembrane space and the mitochondrial matrix. So if you have a lot of protons or hydrogen ions on one side and you have a membrane that is actually like barricading them, there's going to be like energy that's actually going to produce in this particular energy, lot in this particular space here. Lots of hydrogen ion would want to get out of there. And the only way they could get out of there is when there is something that would allow them to move across. And that something happens to be an ATP synthase. This is your ATP synthase right here. And that is your ATP synthase right there. The ATP synthase opens up, allows, allows the hydrogen ions to move into it. And that would allow this ATP synthase to convert ADP plus P into ATP. Could you imagine how many ATP? For every 3 NADPH, you can actually produce 3 ATPs. For every 2 FADH, you produce 2 ATP. That's a lot of ATP. Probably a total between 36 to theoretically 36 to 38 ATP, or depending on what kind of formula you're actually using, because there are scientists who uses the norm that for every NADH instead of 3, there's only like 2.5. Some of them produces only 2.5 instead of 3. And for every FADH2, some of them uses like instead of 1.5 uses 2. So depending on which particular paradigm you're on, the oxidative phosphorylation is actually producing a lot more of ATP than Krebs cycle and glycolysis. So let's take a look at this again. So protons flow down here. We have got protons right here that is highly concentrated in the intermembrane space, and then the mitochondrial matrix. So protons flow down their electrochemical gradient, okay, through the stationary, what they call it stator or ATP synthase. Each hydrogen ion binds to the subunit of the rotor of the enzyme, causing it to change in shape and making the rotor spin. The spinning of the rotor turns the connecting rod. And as the rod spins, this is actually the rod right there, as the rod spins, it activates catalytic sites in the knob that joins the phosphate to the ADP to form one ATP. Pretty much, in a nutshell, that is actually how the ATP synthase will form an ATP. So we're done with the discussion of the um, cellular respiration. Let's take a look at some homeostatic imbalances. Cyanide. We know cyanide, the cyanide gas that is used in gas chambers. How do they kill us? Well, the cyanide disrupts oxidative phosphorylation in our body. And it does that by binding to cytochrome oxidases. It blocks the electron flow from complex 4 to oxygen. Poisons that are oxygen poisons that are called uncouplers will um, will actually abolish proton gradient by making the inner mitochondrial membrane permeable to hydrogen ion. So that means you're not going to have any gradient, and therefore you are not going to be producing much ATP. Although the electron transport chain continues to deliver electrons to oxygen at a very fast pace, the oxygen consumption rises but no ATP is produced. And that's how cyanide kills us. So let's take a look at the summary of ATP production. The energy used at the rest averages about 100 kilocalories per hour, which is equivalent to about 116 watts, slightly more than a light bulb, what a light bulb uses. And it places a huge demand on the mitochondria for energy production. Of the 686 calories in 1 mole of glucose, 262 calories are captured in the ATP, with rest lost as heat. A lot of them is actually lost as heat. This translates to about 38% efficiency in usage of fuel available. That's not really very good, 38%. Wow. I remind you guys, our machines now are only around 10 to 30% in their efficiency. That is something that you need to think, something for you to think about, that our body is more efficient than machines. And we think that machines are more efficient than us. because we see them and we don't see the reality that's inside our body. We are efficient. So let's take a look in a nutshell the energy yield in cellular respiration per molecule of glucose. From 1 molecule of glucose, cellular respiration produces a theoretical maximum of 38 ATP, 10 NADH, 2 FADH. However, the actual ATP yield is closer to 30 to 32 ATP due to some energy loss of transporting the cytosolic NADH to the mitochondria. Here's a summary of the stage. Break it down. First, in glycolysis, you have a net gain of 2 ATP, 2 NADH. That's all you get. Then in the pyruvate to acetyl coenzyme conversion, this is the transition reaction. You have no ATP produced, but you have 2 NADH. This is high energy molecule. Then you have in the citric acid cycle, this one produces 2 ATP per molecule of glucose, 6 NADH, and 6 FADH2. And in the oxidative phosphorylation, all of this high energy molecule NADH and FADH2 will now be converted into ATP. This stage produces most of the ATP using NADH and FADH2 from the previous stages. The total theoretical yield from NADH and FADH2 is about 28 to 30 ATP molecules, or depending on the theoretical maximum in the calculations, this is actually going to be like 38, 36 to 38 ATP. No more than 38. Wow, that was really a lot of biochemistry. And we're not going to leave biochemistry yet. We're going to have to make sure that we turn into more of the chemicals in that is involving carbohydrates. We're still discussing the metabolism of carbohydrates. And this is what you call glycogenesis and uh, glycogenolysis. Glycogenesis is the synthesis of glycogen from glucose, and it occurs when glucose supplies exceed the demand for ATP. So glycogenesis happens in the liver whenever you have too much glucose, and some of the glucose will have to be stored in the liver as glycogen. Glycogenolysis, on the other hand, is a breakdown of glycogen in the liver to release glucose, which is pretty much stimulated by low blood glucose. And when we are hungry, that particular hunger is actually a stimulus coming from our hypothalamus, which is the hunger and the seat for hunger and satiety. So our hypothalamus is actually the one um, um, creating the signal to make us feel hungry by lowering our blood sugar, or if our blood sugar is low, because we have already consumed all the glycogen that we have just eaten for the day, and therefore there are no more glucose around. Therefore, we need more glycogen in the liver. So glycogenolysis will need to happen, break down glycogen to release more glucose so that you can have, again, you can have energy because your energy is coming from glucose. So gluconeogenesis, on the other hand, is a process of forming new glucose from non-carbohydrate sources. And gluconeogenesis happens only in the liver. It occurs in the liver, and glucose can be formed from glycerol and amino acids when blood glucose level is so low, it drops. This is a protection against damaging effects of low blood glucose level or hypoglycemia. And this is especially important for the nervous system because the nervous system is pretty much like in demand for glucose. Okay, um, our nerves and our brain actually is pretty much in great need for glucose, for their source of ATP. So let's take a quick summary of the carbohydrate reaction. First, glycolysis. It's a conversion of glucose to pyruvic acid, breakdown of glucose. Then you have glycogenesis. It's actually the polymerization of glucose to form glycogen. Glycogenolysis is the hydrolysis of glycogen to form glucose monomers. This is when you break down glycogen to form glucose, smaller units. And gluconeogenesis is the formation of glucose from non-carbohydrate precursors. These are just the words that you need to remember for your exams. Lipids. All right. We're getting there. We're getting closer to the end of for the end of the show today. Lipids pretty much are the fats and oils. And again, for an adult that would pretty much like be eating 2,000 kilocalories per day, about 77 to 88 milligrams of lipids daily should be part of the diet. Okay, but about 9 grams of it should be omega-6, 2 grams should be about omega-3, less than 25 grams will be saturated fats. Okay, and the rest of them, okay, will need to be the non-saturated fat and the other important oils. Lipids provide a greater energy yield than glucose or protein catabolism. And you should know that um, the ATP that we get from glucose is nothing if you compare it from the ATP that we get from the acetyl that we get from lipids, the triglycerides. The triglycerides have plenty of acetyl, and you form a lot of acetyl coenzymes there, and then a lot of ATP for the acetyl group from the acetyl group of the lipid, a broken down triglyceride. Fat catabolism yields 9 kilocalories per gram versus 4 kilocalories per gram from carbohydrates. So that's actually more than half of what the carbohydrates can give. Most products of digestion are transported in the lymph as chylomicrons and then hydrolyzed by endothelial enzymes to fatty acids and glycerol. And you have two types of fat. You have the saturated fat and the unsaturated fat. Saturated fats are solid at room temperature and can raise bad cholesterol levels, and they increase the risk of heart diseases. These fats are commonly found in animal products like meats and dairy, as well as processed food. The unsaturated fats are healthy fats found in foods like nuts and avocados, known to improve heart health, and these fats remain liquid at room temperature and helps lower bad cholesterol levels in the body. Let's take a look at the oxidation of glycerol and fatty acids. Only triglycerides coming from the fats, dietary fats, only triglycerides are routinely oxidized for energy. And these two building blocks of the triglyceride, when you break them down, you get fatty acid and glycerol. These are oxidized separately. Glycerol breakdown pretty much is broken down into glyceraldehyde phosphate, the same as glycolysis, which is then entering the citric acid cycle. So glycerol enters the citric acid cycle. ATP yield is roughly half that of glucose because glyceraldehyde is only half a glucose. So it yields about 15 ATP per glycerol. The fatty acid breakdown, pretty much the chains are broken down into two carbon acetic fragments. So it's broken down into twos, and the coenzymes FAD and NAD are reduced in the process. In the process of doing that, you produce a lot of FADH2 and NADH And this is actually highly energetic. This reaction is highly energetic when you break down fatty acids. Acetic acid fragments fuses with coenzyme to form acetyl coenzyme and then enters the citric acid cycle. The reduced coenzymes enter in the electron transport chain. This is referred to as beta oxidation because two carbons are broken off the fatty acid chain, allowing the third position carbon to be oxidized. Here's actually your triglyceride. The two building blocks of triglycerides are oxidized separately. Euglycerol pretty much enters into the glycolysis and results in the production of pyruvic acid. And it will form acetyl coenzyme. In the same token, your fatty acid will be broken down, will undergo beta oxidation in the mitochondria, and still forms acetyl coenzyme. This acetyl coenzyme is now going to get into the citric acid cycle. But one triglyceride can actually form, what do you call this, much more ATP than carbon dioxide. I mean, not carbon dioxide, carbohydrates. Here's again another way of looking at it. You have your triglyceride broken down into glycerol that goes into glycolysis. Your fatty acid undergoes beta oxidation, forms acetyl coenzyme. All of those acetyl coenzyme now gets into the citric acid cycle. Very much. Some of them undergoes ketogenesis and produces ketone bodies. Okay, and some of them, certain coenzymes, certain amino acids, pretty much certain amino acid is also broken down to produce to get in to become acetyl coenzyme and pyruvic acid, and they all get into the citric acid cycle. And eventually, all the byproducts, high energy byproducts, is going to be used in the oxidative phosphorylation in producing more ATP. ATP, carbon dioxide, and water. So, a quick summary of lipid reaction. Beta oxidation converts fatty acid to acetyl coenzyme. Lipolysis breaks down lipids to fatty acids and glycerol. Lipogenesis forms lipids from acetyl coenzyme and glyceraldehyde 3-phosphate. So, you have heard of ketone bodies, and this is probably not the only time you're going to hear ketone bodies because you will hear them again when we get into the study of the kidneys. Because ketone bodies is actually one of those things that you could find in um, in the urine, and it's actually um, a way of pretty much like looking at whether you are healthy or not. And this is actually one of them. When is ketone bodies going to be found in your urine? So accumulation of ketone bodies in the blood can lead to what you call ketosis. And very common in starvation. So you know that if you have ketone bodies in the urine, then you know that that particular person must be dieting unwisely or unnecessarily, or that person has diabetic mellitus or starvation, because this is actually where ketosis happens. When you are starving because you are dieting, or you are really starving, and then when you have diabetes mellitus. Ketone bodies are also, and the ketone bodies are excreted in the urine, and they are acidic. So a buildup of these molecules can lead to what you call metabolic acidosis, which can dangerously um, make your the pH level go down. The patient's breath this time can smell fruity from the vaporizing acetone. Also, the breathing becomes rapid as the lungs try to release carbon dioxide to raise the pH. And these are the three types of ketone bodies: the acetoacetate, the beta hydrobutyric acid, and acetone. And yes, we do produce acetone in our body. So, one last question before we end. how do lipids help in the synthesis of structural materials? Well, lipids may play many roles in structural materials. Phospholipids are used for cell membrane and myelin. Cholesterol is used for cell membrane and steroid hormone synthesis. And the liver also plays a major role in lipid metabolism because it synthesizes transport lipoproteins for cholesterol and fats. And it synthesizes cholesterol also from acetyl coenzyme. It uses cholesterol to form bile salts. And pretty much we are at the end, finally. And we're now going to get into the second part after this.